Hello everyone and welcome to today's live broadcast, Emergence of Clinical Proteomics, Identifying and Developing Actionable Protein Biomarkers for Use in Clinical Research, presented by Dr. Todd Hembrough, President of Proteomics at NantOmics, and Dr. Mary Ann Vogelsang, Manager of Assay Development at NantOmics. I'm Susie Valdez of LabRoots, and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots and sponsored by Thermo Fisher Scientific. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on that green Q&A button located at the lower left of your presentation window and type your questions into the box that appear on the screen. We'll answer as many questions as we have time for at the end of the presentation. Also, please notice you will be viewing the presentation in a slide window. To enlarge that window, just click on the screen icon located at the lower right. If you have any trouble seeing or hearing the presentation, click on the support button at the top right of your presentation window or use that Q&A button and let us know you're having a problem. This presentation is educational and offers continuing education credits. Please click on the bottom button in the left corner and follow the process for obtaining your credits. I'd now like to introduce our two presenters. First up is Dr. Todd Hembrough. Todd is the President of Proteomics at NantOmics, where he is overseeing R&D, operations, and the Clinical Proteomics Laboratory. Todd obtained his Ph.D. in Biochemistry from the University of Virginia, studying the role of proteases in tumor growth and invasion. Prior to NantOmics, Todd was a leader of the translational oncology efforts at EntreMed, focusing on defining druggable molecule pathways and inventing new drugs to target these pathways. Todd later became the Chief Scientific Officer and Head of R&D at OncoPlex Diagnostics and Expression Pathology, where he helped lead the development and commercial launch of the first quantitative proteomics assay performed on formalin-fixed, paraffin-embedded tissue. Joining Todd today is our second presenter, Dr. Mary Ann Vogelsang. Mary Ann leads the assay development team and drives the implementation and delivery of NantOmics mass spectrometry MS-based assay development pipeline for both R&D and clinical proteomics laboratories. Mary Ann earned her Ph.D. in Biochemistry and Molecular Biology from the University of California, Los Angeles. Prior to NantOmics, Mary Ann established a CLIA-compliant laboratory that offered both ELISA and mass spec-based clinical assays. She has over a decade of experience in translational medicine utilizing the mass spectrometry platform and spent a significant part of her career as a scientist developing workflows that facilitate proteomics biomarkers discovery and validation processes at Thermo Fisher Scientific Brim Center. I will now turn the presentation over to our presenters, Dr. Todd Hembrough and Dr. Mary Ann Vogelsang. Welcome, both of you.
Thank you very much, Susie. It's a pleasure to have an opportunity to present today and to really dig into how clinical proteomics is growing and how we're defining new biomarkers and helping to set the stage for the emergence of these biomarkers into helping to understand cancer. So, quick disclosures. Mary and I are both employees of NantOmics. At NantOmics, we really focus on three things. The first is we have a CAP-CLIA clinical lab where we perform clinical proteomics and genomics testing of patient samples. We run whole genome sequencing, we perform RNA-seq, and we perform targeted proteomics. And the idea here is really to get as much information as possible about the patient's tumor so that we can understand the biology of that tumor. Second, where genomics is a maturing space, proteomics is really nascent. So we want to perform as many studies as we can to define the utility of these proteomics assays. And in order to do that, we have efforts in discovery where we're looking to define and discover new targets and new pathways. We have a focus on clinical studies where we do analysis of randomized trials looking in a prospective, retrospective way to test biomarkers that we know we have to see what their utility is in those studies. And then we also perform prospective studies where we're helping to define populations where we think the biomarkers are important. The final area that we focus on is to work with our collaborators and clients in the pharma space to develop clinical biomarkers to help in a companion diagnostic setting. So I'd like to turn it over to Marianne, who's going to describe the technology about how we do it, and here is Marianne.
Thank you, Todd. One of the core goals of what we do here at NantOmics every day is analyze the proteome of tumor tissue. And it all starts with a tumor biopsy. As you look here in this slide, step one, we receive samples and we prepare them on director slides. And then step two, we then laser microdissection. A critical part of this step is the markup from the pathologist that only marks the tumor region, and only the tumor region is dissected and collected. And as you see in step 4, it's a proprietary liquid tissue prep process that liquefies the tissue, and then we digest it in preparation for the mass spectrometer. And so what our platform utilizes is a triple quadrupole mass spectrometer, and then through analysis of that data, we're able to deliver quantitative information at the atomole level for every tissue that runs through our laboratory. And using this platform, we've been able to develop a massive multiplex proteomic panel of clinically relevant targets. A lot of them are relevant for chemotherapies, immunotherapies, as well as agents in development. And today's presentation, I'll focus a little bit later on the development of the HER2 assay, and then Todd will highlight some of our research findings with the TUB3 and TIMP biomarkers. And so, as I said before, the core goal of what we do is analyzing the proteome of tumor tissues, and it all starts with that precious sample. Samples are precious and often limited in size and scope, and so it's critical that we're able to measure as many targets as we can within a single run, because often we're only given one shot, and we get enough sample for one injection. And so a lot of what we do is build methods, multiplex methods of all the targets, and we measure them simultaneously. And we have the power to measure anywhere between 70 and 100 targets at any one time within an hour. And so one of the critical components of this process is using isotopically labeled peptides. And from there, we're able to accurately identify and quantitate our biomarkers of interest and give out quantitative readouts using that ratio, as you can see here on the the right side of the panel, the relative ratio between the endogenous and heavy labeled peptides. Now, in this next slide, I'd like to just sort of highlight our development process. I won't go over in detail every single step. I sort of want to just highlight what we do differently here. We do have a strong molecular oncology knowledge base that we utilize in-house, and we also have a genomics company as part of a division within NantOmics. And together with strong oncology knowledge as well as genomic data, we're able to build candidate target lists. Something else that we do differently is utilize recombinant proteins. Because a lot of what we do and develop starts off with a literature knowledge or in silico understanding, we want to be able to understand how those proteins and those targets behave in the mass spectrometer. So we start off with a recombinant protein, digest that, get characteristics of how that behaves on the mass spectrometer, and then we order the heavy labeled peptides that we think will be robust throughout the validation process. And then as we screen in early development stage as well as final validation stage, we utilize the internal standards in all of our tissue samples and in every run. And you see right here, as you can see at this last stage, all of the processes that we go through monitoring the heavy and light peptides will go through our validation process, which I'll be explaining to you today, and try to characterize that performance. So much of what I'll do today, I'm going to make a couple assumptions of the readers today, that you already have your chromatography selected, but just to highlight what's important in picking out the right chromatography, you want to make sure that you have distinct peak shapes and ideal separation, as well as robust and stable retention times. Additionally, the use of that stable isotope, I can't stress enough, as being valuable for this entire validation process. And so let me go over a couple parameters that you need to keep in mind in assessing the performance of your targets, and that's in the mass spectrum signature, or your peak shape. And so you want to make sure that your retention time is stable from your isotopically labeled peptide and is in alignment with your endogenous peptide, of course, but you really need to make sure that that retention time from run to run is stable, and that enables the multiplexing and the scheduling capabilities that you have within the mass spectrometer. And also, you want to, I mentioned before, we calculate the amount of the protein based on the light to heavy ratio. You also need to be mindful of the isotopic ratios of the transition ions within each peak. Now, what we do a little differently is we build methods that can sustain different tissue types, and often you can see different transition ion ratio performance based on those tissue types. And so, excuse me, sorry about that. You need to make sure that your transition ion ratios are stable across any matrix and any tissue type, and that's what we're doing here. In order to understand the complete process of how to validate your targets, I'm going to refer you to a handful of CLSI guidelines. CLSI is a Clinical and Laboratory Standards Institute. They've published numerous guidelines, and I'd like to call out C62A and C50A as guidelines that are invaluable when you should refer to when you are validating your targets, because they are specifically mass spec focused. And when you do read through these guidelines, you'll notice that there is a consensus between them, and that outlines 8 specific steps in analytically validating your methods. They range from assessing specificity, accuracy, precision, and all the way down to robustness. And I'll go through a couple of these. Okay, and in these slides, in this next slide, I'd like to go over just a few highlights of those 8 processes. I'm not going to read the definitions out loud for you. I'd like you to come back and read these. But however, I'd want to call out 2 specific steps. It's the specificity step and the precision. In order to know that you're undoubtedly measuring the analyte of interest, you need that internal standard. And it'll just help you measurably when you're looking at different mediums, whether it's serum and plasma, and in our case, different tissue types that cover different indications, as well as precision. The precision step, measuring your samples over a period of time repeatedly, 21 days, 30 days, or years later, you want to make sure that your assay is robust and that you have the same measurement day in and day out. The next few steps, limit of detection and limit of quantitation, sort of define and highlight the sensitivity of your assay. And in our case, working off of 1 microgram of tissue, that couldn't be more important. However, because we run a large operation, I'm also going to ask you to be mindful when you really want to take your discoveries to the next step, to look at ruggedness and robustness. You want to make sure that, we make sure at least, that our assays perform across different instruments as well as across different tissues. And that's where, when an assay is robust, we do see a difference in how they perform across different tissue types. And we're always mindful of that and document it in our validation process. Now, let's go to the fun step. I actually like this slide. This slide here, slide 14, describes and highlights how we validated the HER2 assay. And again, in step A, we did start off with a recombinant protein and assessed 3 of the peptides, and the peptide ELVS was the most robust and the best performing peptide. And so as you can see here in panel A, it is the MS2 spectrum of that heavy peptide. As well as in panel D, I'm calling out sort of the transition ions that we're using to quantitate. And the top half is the light, and the bottom half is the heavy peptide. And so this assay performs well over a wide dynamic range, as you can see in panel B. it gives us a linear performance from 150 attimoles to 25,000 attimoles. And in panel E, this is one of my favorite parts of the figures, because it highlights how robust and how rugged our assay is across different instruments. If you look at the y-axis and the x-axis, we're comparing two different instruments. And if you're looking at the difference in color, we're also showing how well our assay performs under different tissue types. We have breast cancer as well as gastroesophageal cancer. And when looking at panel F, you can see how important precision is. And often the mass spectrometer lends itself to the ability to build robust assays time after time, as well as the use, in our case, of formalin-fixed tissue. We're able to see a strong performance of the assay across 1 year apart. So you can see the x-axis is a freshly prepared tumor tissue sample, as well as one from a year ago on the y-axis, and we still get a high degree of agreement. And I'd like to refer you to our paper last year in gastric cancer for more details. So we do perform all the steps for every assay that comes through our lab, but one of the powerful components of being able to build assays on the mass spectrometer is this idea of multiplexing. So we have to run as many targets as we can, 70 to 100 targets at one point. However, every time you do add another target into your method, you need to be mindful that you're not compromising the ability and performance of that assay you designed before. So you want to be mindful of your peak shape, points across the peak, and signal to noise every time you add to your methods and when you're validating your assay. It's always going to be a balancing act between throughput and sensitivity. And initially we started out, sensitivity on 1 microgram of tissue is essential. However, because we know that we need to be able to measure as many targets as we can across indications, throughput is equally valuable in our case. And I challenge you to think about these components as you're developing your own assays in your laboratory. And with that, I'd like to hand it over to Todd, who will talk about the clinical utility of these assays in our research laboratory.
Thanks a lot, Marianne. That was great. And from my perspective as a molecular oncologist, what we've always looked at is that the mass spec is this really powerful tool, and we can get all this information. And as she said, the critical point is validate the assay so that you can trust the data, so that you can run it again and again, year in and year out, and you can always trust the data. And I think that what Marianne has shared is just a snapshot of the really strong, detail-oriented focus on making sure that it's being done perfectly. So then the challenge for us is, does it matter? Within the oncology space where we work, there's many ideas that fail to be proved scientifically, and logic and oncology don't really overlap. So what we did is we started at the beginning. We started with an assay that everybody understood within the oncology space, HER2. HER2 is a kinase receptor. It's a driver in breast cancer, and it's a target that there are drugs in the clinic, and everybody knows how to use the data. So we started by comparing how mass spectrometry compares with the standard assay, which is called immunohistochemistry, where antibody staining is performed on tissue. So in this slide in the middle, you can see these boxes with IHC. We received 19 tumor samples from MD Anderson, and we reprocessed them to show that each of those samples was indeed highly staining for HER2 by immunohistochemistry. Now, in the clinic, this is a diagnostic test that's performed to tell the physician to treat this patient with HER2 therapy. Now, what we really wanted to know is how well was that assay performing? And we were surprised to discover that when we took 19 samples that were all identical, what we saw was a 100-fold range of protein expression within this population of seemingly identical immunohistochemistry. And so you can see that the assumption that we have is we believe that the different parts of this population will actually perform differently in the clinic. And so in order to test this, we took this and collaborated with a laboratory in Spain, and we looked at their experience with FISH, with immunohistochemistry, and with what we performed, which was targeted proteomics. And again, where the clinical research folks who focus on FISH and IHC, they see these things as the same. They see HER2 FISH as positive, they say IHC3+ as positive, and again, what we see is this vast dynamic range of expression of HER2 protein. Now, again, the punchline here for us is we believe that the patients who have lots of protein and have no protein ought to behave differently when they're treated with therapy. And so we took this population and looked at how the patients responded to the drugs. Now, this is a retrospective study. We got clinical research samples from the Vall-d'Hebron Institute of Oncology in Spain, and we performed all of this analysis long after the patients were treated. So this is the canonical clinical research study where we're looking at performance and we're trying to associate how the expression of protein translates into outcome in these clinical research samples. And as you can see on this slide, I'm sort of selling the punchline, which is we found quantitative proteomic cutoffs which were predictive for FISH amplification at 740 and are predictive for benefit on the drug, which I'll show you on the next slide. So in this population of breast cancer patients who were treated in the adjuvant setting, when we separated the population based on this cutoff of 2,200 nanomoles, what we saw is that those patients had a demonstrably different outcome. The patients who had lots of HER2 responded very well to HER2, and in fact, none of them died in this study, whereas the patients who had less or very low levels of HER2, as you saw on the last slide, performed worse. Now, this corresponds to what we hypothesized, which is patients with lots of HER2 protein should be responding to the targeted therapy better, and patients with very little HER2 shouldn't be. And we saw the same phenotypic response in the metastatic setting. So these are the two key settings for breast cancer. Adjuvant setting where there's surgery and then treatment, and metastatic where the patient shows up with metastatic disease. And again, in both cases, in the metastatic setting, this cutoff was able to separate the patients who performed well in the top curve versus the patients whose tumors didn't respond to the therapy and who sadly perished more quickly. Now, the exciting thing about this from the mass spectrometry perspective is, in looking at these patient samples, and again in the clinical research setting, there's no other way to analyze these patients and see this data. When one uses the other standard ways to measure HER2 in these patients, either FISH, IHC, gene copy number, they don't see this separation. So what we have with mass spectrometry is a very powerful tool to look at what's really going on biologically in these samples and understand how the biology differs between patients that look the same with other technologies. Now, one of the assumptions that we have is that biology drives cancer. So what we know is that HER2 is expressed highly, it's amplified in breast cancer, but it's also highly expressed in gastric cancer. So the hypothesis when we stepped into this next study is that the behavior of HER2 in both diseases should be the same. It's a driver, it's an oncogene, and it's targetable. So this is a second clinical research study where we took patients who had been treated in the past, they had been part of a clinical trial, and what we're doing is we're analyzing this tissue in order to understand how HER2 expression is relevant to the response that the patient showed with these therapies. So again, in this slide you can see the distribution of IHC. As we showed you in the last slide, in the 3+, which is the expression level where these patients are being given therapy, you can see that once again there's an extremely broad dynamic range of expression, including patients who actually have no protein expression, where their immunohistochemistry may in fact be spurious. And so when we look at this population, what we see is two subsets of patients. Patients who express lots of HER2, but because their immunohistochemistry was low, they wouldn't have been recognized as expressing lots of HER2. And then a population where their immunohistochemistry says they have a lot of HER2, but what we see is low expression of protein. So as we showed in the breast cancer study, our hypothesis is that this patient population, number 2, is a population that when they are treated with HER2 therapy will be less likely to respond to the therapy. So when we did this study, we performed what's called a prospective retrospective study. We defined the cutoffs, we tested the cutoffs in order to see how this population performed using our diagnostic. And so you can see that the cutoff here that was tested was 1,825 attomoles of HER2. And this was a randomized clinical trial that was performed. We got the tissue, and then we processed it. And so what I want to highlight here is, on the right-hand side, is the fact that this is overall survival, and that we have 3 different cohorts within the study. There's a population who are treated with a HER2-targeted drug called Herceptin and chemotherapy, and those are the blue and the greenish curves. And then there was a population that was HER2 expressing, but it wasn't treated with a HER2-targeted therapy. Instead, it was only treated with chemotherapy. So what you see on the right-hand side is that when a patient expresses HER2 above our quantitative cutoff and is treated with Herceptin and chemotherapy, that those patients in the blue curve respond or perform better under that therapy than patients in the greenish curve who are HER2 positive, HER2 expression low, and treated with Herceptin plus chemotherapy. So right here we're seeing the same biology that we saw in breast cancer, that the more HER2 you have, the better you respond. The less HER2 you have, the less well you respond. But what's most striking about this is the red curve, which represents a HER2-treated population, a HER2 population treated with chemotherapy alone. And what's really important to capture here is that this population of chemotherapy alone and the population of the green curve, which is HER2 treated, but Herceptin treated but HER2 low, was statistically indistinguishable. And what this really suggests is that in this population, based on this, retrospective analysis, is it suggests that there is a population of patients who have so little HER2 that they're doing no better by being given Herceptin than if they were just given chemo alone. So now, as I said earlier, the key underlying hypothesis of what we do is that biology matters, and it doesn't matter where you see the biology. So this is a distribution curve that captures where we see superexpression of HER2 in any indication. Now, right now, clinical testing is limited to breast cancer and gastric cancer, but our research findings here suggest that there are many, many more places where HER2 is overexpressed and may actually be a therapeutic target. And I think from a clinical research perspective, this is information that needs to be translated forward so that people understand that perhaps they should be looking in more places for high HER2 because they will find patients with cancers outside of where they're used to looking who may be benefited by these therapies. And I think this is a really exciting observation with mass spectrometry, and it really just needs to be pushed so that more and more people understand it. And I think that ultimately biology is what matters. And as Marianne said early on, the goal, everything that underlies what we do is understanding the proteome of cancer so that we can understand the biology. And the final piece of the HER2 story is here with bone. Most technologies that are being used in the diagnostic lab struggle with bone metastases because of the decalcification of the bone. And this is a sample that we received in the research lab where it was a bone met, and we were able to analyze it even though it had been decalcified. And this is one of the exciting pieces of mass spectrometry as well, is that the utility of the biomarkers when we develop them, it crosses into all different tissue types. So we don't need to be narrowly focused on, one indication. We can do research, we can do, protein biomarker research anywhere. And this is a case where the other technologies, IHC and FISH, were not able to analyze this tissue. But when we analyzed it in the research lab, we were able to see that this tissue expressed a huge amount of HER2. And so this underscores, again, the need to understand the biology and the need to really, ask the questions, what is driving the tumor, and how can we build protein biomarkers to understand that? So that's a snapshot of the targeted therapy space, and there's lots of exciting efforts going on in the targeted therapy. But we also have as a goal to understand what historically has been called, unlike smart drugs, dumb drugs. So chemotherapy is given to almost every patient, but right now it's not being used in a biologically targeted way. And I think that what our goal with chemotherapy is, can we do research on the potential biomarkers for chemotherapy drugs and understand whether or not there are protein biomarkers which can be informative as to whether or not the tumor might respond or not respond to a specific chemotherapy. And this is becoming more and more important because, as I'm sure you're all familiar with this revolution in immuno-oncology, most chemotherapy drugs have profound impacts on the immune system. They may activate the immune system, or in fact they may, suppress the immune system. And the current method of using chemo without biologically aligning it with the tumor, it has really profound impacts on the ability to take the next step into that immuno-oncology space. And then finally, in the drug development area, one of the exciting spaces that is in development and research is antibody-drug conjugates. And this is an antibody like a HER2 antibody, which happens to be connected to chemo. And the idea of understanding how chemo can be targeted is really to understand how whatever biology a patient's tumor has, we can understand it and then understand what might be good choices for that tumor. So again, from the clinical research side, we start all of our studies with a retrospective analysis of clinical trials. So this is a clinical trial. It was a gastric cancer trial, and it looked at two different chemo regimens, one which included docetaxel, which is a taxane which blocks tubulin, microtubule formation, and another which used a metabolic inhibitor called 5-fluorouracil. Now, the irony is that when they performed this clinical trial, their conclusion was that the trial failed. There was no difference between these drugs. So what we sought to do was to take this study and simply ask the question, can the known biomarkers, the TUB3, which is a tubulin isoform, that have been studied and researched and proposed to be biomarkers of taxane activity, can we look at this research study? Can we test whether or not the expression of this protein, tubulin beta-3, has anything to do with the response of these patients to taxane? And so we were really excited to see that indeed it did. And again, I compared the immunohistochemistry data for HER2 with the mass spectrometry to show that mass spectrometry has a much better range of expression, sensitivity, and I think that the same thing is active here, where tubulin beta-3 has been studied using multiple different technologies. It's only when one does mass spectrometry that one starts to be able to see the granularity of expression that helps us understand what are the biological expression levels of tubulin beta-3 that might be sufficient to decrease docetaxel activity. So what we're looking at here is the two arms of the clinical trial. On the left is the arm which is 125 patients who are treated with taxane plus platinum, and on the right is 122 patients who are treated with this 5-FU metabolic drug. Now, our hypothesis is that tubulin beta-3 is relevant for taxane, and you can see on the left side that when tubulin beta-3 is absent, the blue curve, those patients perform better on this therapeutic regimen than the patients in the red curve who have tubulin beta-3. So this supports the idea that, yes, tubulin beta-3 probably is a resistance marker, and when it's present, the patient is likely to perform worse on this therapy. Now, the right-hand curve, which shows the patients who weren't given taxane, there's no difference between the patient's response to this therapeutic regimen based upon whether there's tubulin beta-3 present or not. So in this retrospective analysis, in a research setting, we would conclude based on this data that TUB3 is predictive of response to docetaxel because it works to separate the population in a docetaxel-treated population, and it has nothing to do with the outcome in the patients who aren't being treated with taxane. So indeed, if you look at this population of patients who have high tubulin beta-3, you can see that the patients who are treated with 5-fluorouracil do far better than the patients who are treated with docetaxel. So from the research and development context, this is the data that needs to be captured in the proteomics space to help people understand the power of using proteomics in tissue and the power of using proteomics to understand tumor biology. Because, you know, these targets have been talked about for years, but just never were able to reach the point where people started to believe the data because of these other technologies. There was always something missing. And I feel like with the targeted proteomics, we're starting to push through to where accurately measuring, you know, with very, very high specificity, we're starting to see these profound differences in these populations. So I want to talk for a couple more minutes about the other arm of that trial. So this is a biological pathway it results in the synthesis of DNA and RNA. And so there's multiple different enzymes that are responsible for this pathway. So what we sought to do was to build assays for all of these targets, to test them all, and to see whether or not the 5-FU arm was in fact impacted by the expression of any of these targets. And indeed it was. And so this is the taxane arm, which you saw on the last slide, where we showed that tubulin beta-3 was a predictive biomarker, and the 5-FU arm on the right. Now, the 5-FU arm, if we look at this enzyme called TIMP, we see that in the presence of TIMP, in the red curve, these patients are responding better to 5-FU than patients who don't have TIMP. And TIMP is an enzyme which activates 5-FU to become incorporated into DNA and RNA and cause DNA damage. So again, taking another look at this research study with our proteomics tools allows us to see perhaps what can't be seen using other technologies. And when we do this, all of a sudden we see that yes, you know, tissue proteomics is a critical tool to understand the biology of these tumors. And as we go forward, I think that the critical goal is to continue to prove and demonstrate that the assumption that biology matters is true. These are tumors that have different biology that's impossible to see without using tissue proteomics. But once one can see the difference in biology, one becomes cognizant of and can understand how different drugs can target different pathways in those tumors. And as I said, I mean, the whole goal here is, you know, take these research studies, understand the biology, and then move forward to share this information so that more and more people understand the power of tissue proteomics. So with that, I will summarize, and I think that the exciting thing for us is how we've taken a very well understood mass spec platform that is one that many, many folks are comfortable with, and we've utilized standard assay development tools, validation tools, things that everybody should also be familiar with, and we try to apply them in a different way. We try to say, look, we know all this information is locked up in the formalin-fixed paraffin-embedded tissue. Let's find a way to get that information out. Let's find a way to interrogate tumors to better and better understand the biology, and then let's find a way to test our hypotheses on these research tissue sets so that we can understand whether or not these proteomics tools have a role in defining the biology of these tumors. And with that, I will say thank you very much for your time. Thank you for your patience, and we would be happy to answer any questions that you have about the assay development, the validation, or indeed the research oncology data that I've shared today.
Thank you, Dr. Hembrough and Dr. Vogelsang, for your informative presentation and for your important research. To our audience members, we will now start the live Q&A portion of this webinar. If you have any questions you'd like to ask, please do so now. We would appreciate your participation also in 2 upcoming polls. You will see them pop up on your screen, and we encourage you to respond by clicking on the answer that best reflects you. So let's begin and take a look at our audience questions already coming in. Our first question is a two-part question. What do you think is or are the largest barriers in generating broader adoption of MS-based proteomics in the clinical space? That's the first question. And how are you making the case for the MS platform to replace IHC, ELISA, or other orthogonal assays?
Well, thank you for that question. I think it's an important question that all of us in the field are considering. And I think one of the main barriers is probably cost, even above, trained personnel, because we have many people that are trained and able to use the mass spec. I just think it's not necessarily as easy yet for users in the clinic. However, I think cost is the largest barrier to entry for the mass spectrometer into the clinical space. But I think multiplexing our assays give us a better shot of having a better return on our investments and bringing the cost down per test in the clinic. Thank you for that question.
And thank you, Dr. Volgosing. Here's our next question. Can these same principles to developing an assay and ascribing utility to an assay be done for analysis of blood? That's the first part, another two-part question. Are there any challenges or special considerations that are applied to analysis of blood using quantitative SRM assays?
Thank you again. Much of the field actually does work in the blood space primarily. I think NantOmics is pretty unique in that we're actually working with tissues. So yes, these principles can be applied to blood and often are in research. And as far as the technicality and considerations when you're applying these methodologies and validating assays in blood specimen, it's the same. You treat it, the interference that you might see in any of your mass spec signal would be treated the same as if they were blood or from a tissue. It's still the same technical validation process that you would run through. Thanks. These are fun questions.
And we have some great questions coming in from our audience members today. Our next question, you mentioned global proteomics on an intro slide, but don't discuss it in the rest of the slides. How have you incorporated global proteomics into your approach?
Oh, thanks. Yeah, we did. We have started to build a global proteomics discovery department here, and a lot of what our assays have built on historically were basically targeted proteomics, and so we used a lot of the literature-based and genomic-based knowledge that we already had in-house and built assays around that, and now we're starting to let global proteomics guide our next set of assays that we're developing.
Thank you.
Thanks.
Have you gotten any examples from any type of brain tumor?
Oh,
really
from brain? Oh, yeah. We work with all types of specimen in our research and in our clinical laboratory. And so, yes, we have.
Okay. Thank you. Our next question, what advice do you have for unknown biomarker ID using mass spectrometry?
So can you repeat that again, please?
Sure. What advice do you have for unknown biomarker ID using mass spectrometry?
So I guess there's a lot of ways you can go about this question. You can, if it's unknown, really in the discovery phase, you're looking at your differential of signals, and you can still carry through this process even without an identification if you want to just go based on the mass spec signal and generate an assay on it. But I do think that if you really want to be an assay in the clinic, you have to truly characterize that target. If I'm answering that question correctly. I don't know if that person can type it in again or follow up, please. Thanks.
Great. And again, thank you, audience members, for your incoming questions. Our next question, how do I find the marker for other types of cancer? Is there a list or a resource of known types of markers for all cancer types?
Oh, I love this question. Thank you. Actually, here at NantOmics, I like to think, or we consider ourselves indication agnostic. So as far as cancers are concerned, we run our entire platform and our entire pipeline of assays on any cancer, regardless of the type, because we think that the biology is characterizing the cancer, not necessarily the cancer itself. And so any given target can express in any given type of cancer. Thanks.
And thank you, Dr. Vogelsang. We have time for one more question, and I want to make sure I give you some time to provide some last words. Which mass spectrometry did you use for your work?
And as I mentioned before, a lot of our, the history and our development of all of our assays so far was built on triple quad mass spectrometers. It's only now recently that we're using Orbitrap-based instruments for discovery efforts, but right now, routinely, when we're quantitating all of our targets, we're still doing them on the triple quadruple instruments.
Great, thank you so
Thanks.
much. And would you like, and I just want to remind audience members that any other questions coming in will be responded via email from our presenters. Did you want to provide any last closing remarks to our audience, Dr. Vogelsang?
Well, I want to thank everyone for participating and listening to today's webinar. It was a really fun experience for me, and I hope I was able to help listeners. And please, if you do have any questions, Todd and I would be happy to follow up through email. Thanks.
And thank you very much. And thank you, Dr. Hembrough, as well. I'd also like to take the opportunity to thank LabRoots and Thermo Fisher Scientific for making today's educational webcast possible. Before we go, I want to remind everyone that today's webcast will be available for on-demand viewing through May 2018. You'll receive an email from LabRoots letting you know that this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now, and thank you for joining us. We hope to see you again soon. Goodbye.